Okay, so this is a good point to mention the two different types of twins. So there's monozygotic twins and dizygotic twins. So in terms of monozygotic, if there's zona pellucida damage, which causes cell separation, then you'll have genetically identical monozygotic twins. And this is pretty rare. It happens 3 in 1,000 births, and they'll share a placenta. Dizygotic twins, however, will have separate eggs and therefore separate placentas, and this is slightly more common, 7 to 11 in 1,000 births, and they'll be genetically different, and it's increased with maternal age and IVF. One of the things that can go wrong if you have a twin is to do with blood flow. So if blood flow is shunted, which means like a bypass, then there might be less blood going to one of the twins, which will result in developmental defects. In terms of clinical application, the fact that the fertilized egg is free living at the early stages means that, well, first of all, IVF treatment can occur, as eggs can be fertilized in vitro and implanted back into the uterus to develop normally. Also, low sperm production can be countered by either concentrating the sperm into a drop or inserting the sperm into the egg via microinjection. You can also do genetic screening, sorry. This allows for identification of genetic diseases and also sex determination. And you can also do genetic engineering because of in the fertilized egg, because by knocking out, for example, by knocking out genes in animal embryos, one can understand their function and thus understand the phenotype of an individual. So after fertilization occurs, from 0 to 3 weeks, as I mentioned right at the start, is the pre-embryonic phase. And I'm just going to start that off in this lecture and carry on in the next. So the pre-embryonic phase results in a blastocyst and a trilaminar disc, which I'm going to talk about later. But what happens essentially is that the morula, which is when the zygote has the cells are large enough to make a separate individual, it differentiates to form the blastocyst. And this is the first specialization and results in two types of cells. The inner cell mass is the embryoblast, and that is going to become the embryo. The trophoblast is the outer layer, shown here. And this forms the life support system, the placenta, which is required as simple diffusion is not sufficient enough to sustain the embryo. So what the trophoblast does is it has a role in implantation of the blastocyst because it's the outer layer. And this means an application of this, it means that IVF can happen and in vitro fertilization can happen. You can fertilize the egg outside of the body and insert it at this stage because the trophoblast is there and can allow implantation into the endometrium of the uterine wall. So just having a closer look at the diagram of the blastocyst, the inner cell mass is the embryoblast and the outer cell mass is the trophoblast. The embryoblast will become the embryo and the trophoblast allows implantation into the endometrium of the uterine wall and it will also produce the placenta. So now to quickly summarize everything that I talked about in this lecture, the three main phases: pre-embryonic, embryonic, and fetal. I gave an overview of what happens in each phase, with the blastocyst and trilaminar disc being created in the pre-embryonic phase, embryonic phase going from 2D to 3D, and the fetal stage being for consolidation and growth. I talked about the difference between male gametes and female gametes and the fact that you have a fixed number of female gametes, whereas male gametes actually increase in number. I gave a quick overview of what happens in fertilizations or ovulation to the oviduct entrance, and then the sperm goes up the peristalsis, then swims to the oviduct entrance and fertilizes the egg. Mentioned the changes in mucus, the reduced mucus due to the hormones in ovulation, the egg structure, as well as the sperm structure and adaptations. Talked a little bit about the differences between the two sets of enzymes and what they're used for, and the changes in the egg layers to prevent polyspermy. Then I mentioned that the zygote becomes a morula when each cell is large enough to make a separate individual. Talked about the differences between the two types of twins and the fact that one shares a placenta and one has separate placentas. Did a bit of clinical application about IVF, genetic screening and genetic engineering. And then we ended on the start of the pre-embryonic phase, which is the creation of the blastocyst with the so far 2 cell layers and what they'll eventually become.